We're on part 5 of the male reproductive system, and we're looking at page 1027 for part 5. A brief note on erectile dysfunction. There are some reasons for erectile dysfunction, some of which might be psychological, some of which might be more physiologically and physically related to age, for one thing. Over the age of 40 or 50 can cause a decline in the function of endothelial nitric oxide and nitric oxide synthase. So there might be less function of that particular system. They talk here about diabetes as being a part of the program, and that in general metabolic syndrome is usually in existence, partly because the endothelial nitric oxide has declined. So another aspect of this is when individuals use serotonin reuptake inhibitors and other types of medications, that sometimes the side effect is to have a reduced erectile stimulation or a reduced flow into the blood flow to cause erection. Okay, so then we go on here to this next stage called spermatogenesis, and here we have a comparison of mitosis over here and meiosis over here. And part of the program that we have to consider is that there's a primordial cell or a stem cell, and that there's going to be replication, and we have to go through, in this particular case of mitosis, a period of dosage regulation in which the cell is going to divide into two cells, and part of that division requires the even division of chromosomes and DNA from the central sort of like equatorial plane here of the metaphase. There's going to be separation there of the replicated DNA molecules to each side, so that each side ends up with the same number of chromosomes as the original primordial cell. Over here we have a slightly unusual situation in which the primordial cell is displaying a tetrad. So there's actually multiple divisions that go on. There is one division that gets us to this next stage in which we have the diploid number of chromosomes, which is in our case 46, and that we had come from a position of 92 chromosomes down now to 46. And then finally there's yet another division in which the gametes each have 23. It's important that the gametes each have 23 so that the sperm that fertilizes the egg then combines to create a situation in which the combined conceptus or zygote will have 46 chromosomes correctly arranged, right, for the development of the embryo and the fetus. Okay, so that's our goal here, is to create a gamete that has exactly 13, all 13 of the particular one each, that is to say, of each of the types of chromosomes, whether it's a sex chromosome, X or Y, or whether it's any of the other autosomes, that there's one each. All of this, I like to call this dosage regulation for mitosis and dosage regulation for meiosis. So you can use whatever terminology you prefer. Okay, and I'm going to press on here. Here they go through a very, how shall I say, carefully captioned situation. I think they might have made a slight mistake at some place later in the book, but here they do seem to have the correct representation of the nomenclature. We begin with tetrads, which is 4N, and then we come down to the regular number, which are in this case 2n, and then we finally end up with the single n number 13, or not 13, 23 chromosomes. So we go from 96 to 46 to 23. Okay. Then we come over here and let's take a look. We're in the wall of the seminiferous tubule. You might remember that we were looking at the wall of a photomicrograph, and here the artist has drawn the picture so that we can make out things much more clearly. There are these primordial cells that are developing through time. Here they've shown us where, in what stage of meiosis things are occurring until we finally get to the development of the spermatozoa. You'll notice that the nuclei are becoming pycnotic and condensed into little sort of arrowheads. This little section here in the neck region is actually completely made out of mitochondria, so very energetic material. Here the mitochondria are absorbing the last of the nutrients. Okay, so also we have a tight junction here between these Sertoli cells, so sometimes called sustenticular cells or sustentocytes. And these cells are actually having tight junctions and creating what is referred to as a testes blood barrier. So these cells are protected from the outside world completely. These are the family jewels, as it were, right? So the little sperm here, or spermatotids they're called, are growing their flagella, and they're absorbing the last of this sort of nutrient mixture right there. And you can see that the head of the sperm is condensed and has this arrowhead shape to it. At the very sort of front end of this, there's actually some enzyme. There's a little encapsulation here of enzymes, so that when it gets to the egg is still, it has a shell. Some people don't realize that the female human egg is actually in a shell. It has a corona radiata around it, and so the egg doesn't hatch until well after it has been fertilized. So we need enzymes to get through that. Here they're sort of backing out of the driveway, as it were, and they actually have no motility at this point, and they are simply being swept along with the testicular fluids in the direction of the epididymis. Okay, so here is our here is a picture of our friends right there, and here is the primordial cell, and you can see how we go through the stages here. Here is the complete sperm. It has a head that has this particular shape to it. It has a little acrosome up here. This section, called the midsection, is filled with mitochondria. It's actually part of the construction, and we have the tail, or the flagellum, right there. So that's that particular development. And then let's take a look here at this. They describe the work of the Sertoli cells here, and the creation of a blood testes barrier. Okay, so that's that particular issue. Then they begin, this is the text part that's better described, I think, in the visual over here on page 1034. We have a relationship then between the pituitary, the anterior pituitary, and also to some degree the hypothalamus, between stimulation of the wall here to create new cells, and also stimulation by way of LH on the Leydig cells to produce testosterone. So there's a double stimulation here, and there's a negative feedback loop. There's a molecule called inhibin, which is feeding back, and then, of course, there's testosterone, which is also feeding back. So LH and FSH are flowing down constitutively, and until they get their feedback, which will then turn them off for the period of time in which they are getting these signals feeding back. That means there's enough going on here, and there's enough testosterone